Hello everyone, if you're a returning subscriber, welcome back. If you're new, welcome to my channel. My name is Dr. Shungo Mupawa. Guys, if you haven't subscribed, please do so. We want to create a community here. Matrix, today we are diving deep into the grade 12 official life sciences paper 2 examination guidelines, your ultimate tool to pass with distinction. The life sciences examination guidelines are not just another boring document. They are actually the key to unlocking what the examiner wants and how to write your answers. From definitions to diagrams, this document tells you what to memorize, what to understand, and what to practice. So, let's break it down together. The first paper 2 topic in the life sciences examination guidelines is DNA, the code for life, which is worth 27 marks. Firstly, with this topic, the examiner expects you to know the DNA molecule in terms of location in plants and animals, and a brief history of the discovery of the DNA molecule, with special reference to the scientists like Crick and Watson, Franklin and Wilkins. You are expected to know the structure of the DNA molecule in terms of its natural structure, which is a double helix, and each strand is being made up of a sequence of DNA nucleotides. The examination guidelines further provide us with 3 components of a DNA nucleotide. My advice is learn the components of the nucleotides as they are given in the examination guidelines. And when asked for the components of the nucleotides in the exam, write them as they are given in the examination guidelines. You are further required to know the structure of the DNA nucleotide using a diagram, as well as the functions of the DNA molecule. Here is the tip. The functions of the DNA molecule are given in the examination guidelines. Also, know them and write them as they are when asked for them. Next, the examiner expects you to know the process of DNA replication in terms of where in the cell it takes place and when in the cell cycle it takes place. You are also expected to know how the process of DNA replication takes place and the significance of DNA replication. Here is the tip. You are not required to know the names of the enzymes that are involved in this process. Also, you are expected to know DNA profiling. Please note, you are not supposed to use the term DNA fingerprinting. Here, all you need to know is how to interpret the DNA profiles. My advice is you use a ruler to match the black bands on the DNA profiles. Some of the uses of DNA are written in a certain way, and I will talk about this in the video that I'll make on DNA profiling. So, stay tuned. The next section under DNA, the code for life, is RNA, which you are expected to know in terms of location, structure, and function. The examination guidelines actually tell us that messenger RNA is synthesized in the nucleus but performs its functions on the ribosome in the cytoplasm, and that transfer RNA is located in the cytoplasm. You are also expected to know the structure of transfer RNA as well as messenger RNA. The examination guidelines also provide the structure of RNA as being a single-stranded molecule consisting of nucleotides. Like DNA, the examiner expects you to know the 3 components of RNA nucleotide, which are the sugar portion, which is ribose sugar, nitrogenous base, and the phosphate portion, as well as the role of RNA during protein synthesis. The last section of DNA, the code for life, is protein synthesis. The examiner expects you to know how DNA and RNA are involved in protein synthesis in terms of transcription and translation. One good thing here is that the examination guidelines provide us with an outline of the processes of transcription and translation. What is given in the examination guidelines in terms of these two processes is exactly what the examiner expects you to know. My advice is, when studying, learn or memorize these two processes as they are in the examination guidelines and write them as they are when asked in the exams. Finally, under DNA, the code for life, the examiner expects you to interpret diagrams illustrating transcription and translation. The second topic in the examination guidelines for paper 2 is meiosis, which is worth 21 marks out of 150 marks. For this topic, you are expected to use your previous knowledge of the structure of the cell, the structure of chromosomes, and mitosis, which you learned in grade 10. You are also expected to be familiar with terms such as haploid, diploid, gametes, and somatic cells, as well as gonosomes and autosomes. Firstly, under meiosis, the examiner expects you to be able to define meiosis and identify the site of meiosis in both plants and animals. Though interphase is not part of meiosis, the examiner expects you to identify the events taking place during interphase. From here, the examiner then expects you to know the events taking place during the first phase of meiosis, which is referred to as meiosis 1, and the second phase of meiosis, which is also known as meiosis 2. The examiner expects you to know the importance of meiosis in terms of production of haploid gametes, having the doubling effect of fertilization so as to maintain a constant number of chromosomes from one generation to the next, and being a mechanism that introduces genetic variation through crossing over, as well as random arrangement of chromosomes at the equator. Please take note that the examination guidelines provide the importance of meiosis, and as usual, my advice is memorize the importance provided and write the points as they are when asked for significance or importance of meiosis in the exam. The other thing you are expected to know is abnormal meiosis in terms of non-disjunction and its effects. Finally, the examiner expects you to know the similarities between meiosis and mitosis, as well as the differences between meiosis and mitosis. The third topic in the examination guidelines for paper 2 is genetics and inheritance, which is worth 48 marks out of 150 marks. Firstly, the examiner expects you to be familiar with genetic concepts such as chromatin and chromosomes, genes and alleles, dominant and recessive alleles, phenotype and genotype, homozygous and heterozygous. You are also expected to know the law of dominance, which is stated in the examination guidelines. As usual, my advice is, when studying, learn the law as it is given in the examination guidelines, and when asked for it, write it as it is. The next section is about monohybrid crosses, where you are expected to know the format for representing a genetic cross. You are also required to state and apply Mendel's principle of segregation. Please note, the principle of segregation is also provided in the examination guidelines. Write it as it is when asked for it in the exam. Also, under monohybrid crosses, the examiner expects you to know the types of dominance, which are complete dominance, incomplete dominance, and codominance. Definitions of these terms are provided in the examination guidelines, and as usual, write them as they are when asked in the exam. Using the prescribed format for presenting a genetic cross, you are expected to solve genetic problems involving the 3 types of dominance discussed and be able to determine the proportions and ratios of genotypes and phenotypes. That is your genotypic and your phenotypic ratios. Sex determination is the next section under genetics and inheritance. Here, you need to know that 22 pairs of chromosomes in human beings are autosomes, and 1 pair of chromosomes are gonosomes or sex chromosomes. You are also expected to know that males have got XY chromosomes, while females have XX chromosomes. And you must be able to distinguish between gonosomes, which are sex chromosomes, and autosomes in the karyotypes of human males and females. Lastly, under sex determination, the examiner expects you to be able to use a genetic cross to show the inheritance of sex. The next section is sex-linked inheritance, where you are expected to be able to define sex-linked alleles and sex-linked disorders. The examiner also expects you to be able to solve genetic problems involving sex-linked disorders such as hemophilia and color blindness using a genetic cross. With blood grouping, the examiner expects you to know that different blood groups result from multiple alleles, which are 3 alleles, and that the combination of these 3 alleles result in 4 blood groups, and be able to solve genetic problems involving inheritance of blood groups. Then we move on to dihybrid crosses, where the examiner expects you to be able to state Mendel's principle of independent assortment. Please note that this principle is provided in the examination guidelines, and my advice is, when studying, learn and memorize it and write it the way it is given when asked for it in the exams. You are also expected to be able to solve genetic problems involving dihybrid inheritance using a genetic cross and being able to determine the genotypic and the phenotypic ratios. Next are genetic pedigrees. Please note that you are not supposed to use the term genetic lineages, though it's given in the examination guidelines. With genetic pedigrees, you are expected to know what it is, and this is given in the examination guidelines, and be able to interpret pedigree diagrams. Then we move on to mutations, where you are expected to define terms like mutation, gene mutation, as well as chromosomal mutation. In terms of gene mutations, the examiner expects you to know the gene mutations in terms of hemophilia and color blindness, as well as chromosomal mutations in terms of Down syndrome. The next section is genetic engineering, where you are expected to define biotechnology and genetic engineering, stem cell research, genetically modified organisms, and cloning. Please note that the examiner does not expect you to know the names of the enzymes that are involved in this process of producing genetically modified organisms. You are also expected to know the advantages and disadvantages of genetic engineering as a process. With paternity testing, the examiner expects you to be able to use blood grouping as well as DNA profiles for paternity. Genetic links conclude the topic genetics and inheritance, where the examiner expects you to know how mutations in mitochondrial DNA are used for tracing ancestry. The last paper 2 topic in the examination guidelines is evolution, which is 54 marks out of 150 marks. This topic takes the largest portion of paper 2. Therefore, if you want to ace your paper 2, make sure you understand this topic. Firstly, you are expected to be able to define biological evolution. Please note that this definition is provided in the examination guidelines. And my advice, as usual, learn it and write it the way it is when asked for it in the exam. You are also expected to know the difference between hypothesis and theory and why evolution is regarded as a scientific theory, which is also provided by the examination guidelines. From there, the examiner expects you to be able to explain the role of fossil record, biogeography, modification by descent, and genetics as evidence for evolution. In terms of variation, the examiner expects you to know the definitions of a biological species and population, as well as the contribution of meiosis, mutations, random fertilization, and random mating to variation. You are also expected to know the types of variation, which are continuous variation and discontinuous variation, as well as examples of characteristics that display these types of variation, for example, height and blood groups, respectively. In terms of the origins of an idea of origins, the examiner expects you to know ideas about Lamarckism, Darwinism, and punctuated equilibrium. For Lamarckism, you are expected to know the 2 laws used by Lamarck to explain evolution and the reason why Lamarck's theory was rejected. With Darwinism, you are expected to know Darwin's theory of evolution by natural selection. Please note that the examination guidelines provide the theory step by step. Therefore, learn it, write it as it is in the exam when asked for it, unless it is asking for application. For punctuated equilibrium, you are expected to know it as the speed at which evolution takes place. And this process is given step by step in the examination guidelines. Learn it and write it as it is when asked in the exam. Then comes artificial selection, which is also known as selective breeding, which involves domesticated animal species and plant species. This is followed by the formation of new species. Here, you are expected to know definitions of species as well as speciation and extinction and their effects on biodiversity. The examiner expects you to know the process of speciation through geographic isolation. Please note, this process is provided in the examination guidelines. Learn it and write it as it is in the exam when asked for it, unless it is asking for application. In terms of mechanisms of reproductive isolation, the examiner expects you to explain a brief outline of reproductive mechanisms that help keep species separate. Then comes evolution at present times, where you are expected to know any one example of natural selection and evolution in present times. For example, HIV resistance to antiretroviral medication. Then we move on to human evolution, where the examiner expects you to know the evidence for common ancestors for living hominids, including humans. Here, you need to be able to interpret the phylogenetic tree showing the place of hominidae in the animal kingdom, and being able to provide the characteristics that humans share with African apes. The examiner also requires you to know the differences between African apes and humans when given diagrams. You also need to know the lines of evidence that support the idea of common ancestor for living hominids, including humans, provided by anatomical features of Ardipithecus, Australopithecus, and Homo. Also, you're supposed to know it in terms of genetic evidence and cultural evidence. Last but not least is the out of Africa hypothesis. Here, the examiner requires you to know that modern humans originated in Africa and then migrated to other continents. You are also expected to be able to provide evidence for the out of Africa hypothesis in terms of fossils of Ardipithecus, Australopithecus, and the Homo species, as well as providing genetic evidence. You also need to know the timeline for the existence of the different species of the genus Homo, the significant features and differences amongst them, and the general interpretation of phylogenetic trees. There you have it. This breakdown gives you a blueprint to pass Life Sciences Paper 2 with confidence. Don't forget to like, share, comment, and subscribe. See you all in the next video. Bye.